understanding how the HIV replicates and understanding how HIV successfully infects a cell tells us what is important for the virus. HIV-1 virions contain an RNA genome that's packaged and protected in a protein shell called a capsid. After infecting a cell, the RNA is converted to DNA, which integrates into the infected cell's genome. So we know that this capsid has to disassemble or uncoat before integration can take place. So we really didn't know where the uncoating occurred, and we just knew that the data that was out there was not convincing to us in either ways. It was thought for many years that the disassembly must occur in the cytoplasm because the capsid was thought to be too big to fit through the nuclear pores. And astonishingly, we found that the capsids were intact in the nucleus. They were imported into the nucleus in an intact or nearly intact way. We know the capsid stably associates with the nuclear envelope, but it stays there for a long time before it gets in. Most of the host cell proteins get in the nucleus within seconds. So we know that the capsid has a difficult time crossing this barrier of the nuclear envelope. Knowing that it happens so late in the process has implications for all aspects of the virus replication cycles. The other surprise was how do these intact capsids get through the nuclear pore into the nucleus? So we think that by understanding how HIV replication occurs in details, we can develop ways to interfere with that replication process and develop new drugs that interfere with HIV and thereby find new ways to control the HIV pandemic.